Right now, bring your fingertip to the inner, touching the opening of a structure. You are telling the opening obstructs confused biologists for centuries the nasolacrimal duct. It routes tears from the surface of your eye down in your nasal cavity. And when you cry, it fills that cavity with fluid. But that fluid, your tears, are not just saline. They are a chemical cocktail designed to do something that no one believed until the 1980s. Physically reduce the amount of pain you feel. There is a measurement you can take, though it requires a single tear and a laboratory. The protein concentration in emotion tears is 24% higher than in tears caused by onion vapor. That statistic comes from Dr. William Frey at the University of Minnesota, who in 1981 collected tear volunteers wanting a sad film and compared them to tears sat onion, and compared them to tears collected when the same volunteers held a fresh-cut onion. What he found inside the emotional tears was a molecular signature that reflect tears did not contain higher levels of prolactin, of adrenocorticotropic hormone, and of a small peptide called leucine-enkephalin. Leucine-enkephalin is an endogenous opioid. It binds to the same receptors that morphine binds to, and it appears in emotional tears at concentration that, if you were at the equivalent amount, would measurably raise your pain threshold. Your body produces 3 kinds of tears. Basal tears lubricate the cornea. You produce about 5 to 1 microliters per minute, just to keep your eyes wet. Reflex tears respond to physical irritants, onion gas, smoke, a stray eyelash. And emotional tears, the third kind, are triggered by the limbic system, specifically the anterior cortex and the irianteri and the amygdala, while direct to the lacrimal gland via the parasympathetic nervous system. When the anterior cingulate cortex activates during tense emotion, it sends a signal down the facial nerve 7, cranial nerve 7, to the lacrimal gland, and that gland immediately begins secreting a different limb. The chemical profile of that fluid is not accidental. It contains pain-modulating molecules, stress hormones being cleared from the blood, and a cascade of neurotransmitters that do not appear in basal or reflex tears at anything close to the same concentration. Now consider the physical path those tears take. They do not just spill over your lower lid and run down your cheek. They also drain through the puncta, those tiny openings. Tears flow in the lacrimal sac, then down the nasolacrimal duct, then down the nasolacrimal duct and empty into the nasal cavity. Your nasal mucosa is a direct portal to the bloodstream, is lined with a dense network of capillaries that absorb small molecules within seconds. But there is a more direct route still. The olfactory nerve endings sit exposed in the roof of the nasal cavity, providing a pathway into the brain that bypasses the blood-brain barrier entirely. This is why intranasal administration of drugs, like naloxone and oxytocin, works so quickly. The molecules travel up the olfactory nerve and reach the cerebrospinal fluid in minutes. The same anatomy applies to the molecules in your own tears. Leucine-enkephalin, the endogenous opioid found in emotional tears, has a molecular weight of approximately 556 daltons, small enough to cross the olfactory mucosa and enter the central nervous system directly. So when you cry and those tears drain in your nose, you are effectively administering a self-produced opioid through the nasal route. The same route now being explored in clinical trials for rapid-acting analgesics. Researchers at the University of California, San Francisco have been testing intranasal enkephalin analogs for migraine relief, and they report onset of action in under 60 seconds. Your body already has the raw material. It simply uses your tears as the delivery vehicle. The effect is not immediate. The tears must accumulate, drain, and be absorbed. This matches the subject experience of crying. The peak of pain relief often comes several minutes after the crying episode, as the chemicals reach their targets. A study published in the Journal of Clinical Psychology in 2008 by the participants who cried during a sad film both emotional and physic reduction in port emotional or tensory treating the chrysler exactly 15 minutes after the crying, stopped not during the crying, but after the nasal absorption had time to occur. That 15-minute delay aligns almost perfectly with the pharmacokinetics of intranasally administered small peptides. But there is a second mechanism operating at the same time. and it involves the vagus nerve cranial nerve X, which runs from the brainstem down through the neck, chest, and abdomen, carrying sensory information from your internal organs back to the brain. When you sob, you engage in a pattern of irregular diaphragmatic contractions that are distinct from normal breathing. Each sob is a forceful exhalation against a partially closed glottis, followed by a rapid inhalation. This pattern sends a powerful signal up the vagus nerve, specifically through its afferent fibers that originate in the diaphragm, the intercostal muscles, and the pharynx. The nucleus tractanas salique in the medullas these signals and initiates a cascade of the period at brain tractable directly mid-brain den grady dem greasen sees involved descending pain modulators. Dr. Stephen Porges, the developer of polyvagal theory, has described how the vagus nerve is divided into two branches, the ventral vagal pathway, which is associated with social engagement and calm states, and the dorsal vagal pathway, which is associated with immobilization and shutdown. Crying, particularly with vocalization, activates the ventral vagal pathway, which inhibits the sympathetic nervous system's fight or flight response. A study from the University in 2014, led by rate variability, Edward Barrow, measured heart rate in vagal tone, the same veloci signators, and found a significant increase in Hayre tone or seen during deep meditation or ECB, ease, mic breathe, E. The participants who showed the greatest increase in vagal tone also reported the largest reductions in pain sensitivity, as measured by a cold pressor test. But here is the key detail. The vagal activation does not occur during the first burst of tears. It takes roughly 1 to 2 minutes of continuous sobbing before the vagal signal reaches the threshold needed to inhibit pain pathways. This is why the most intense emotional crying often feels exhausting, but is followed by a wave of relief the same 15 to 20 minute in nasal route that matches the opioid absorptions from the nasal route. The two mechanisms are not independent. They are synergistic. The chemical opioids from the tears enter the bloodstream and the cerebrospinal fluid. Similantly sees the brain's own opioid system through endogenous releaser trays, erogenous ductal gray, and the rostral the perimedial and the rostral ventromedial medullae. Nadi, you are, in effect, hitting the same pain modulating network from two directions at once. Now there is a third mechanism, and this one is purely mechanical. Your tears, as they flood the nasal cavity, also stimulate the trigeminal nerve cranial nerve, which the trigeminal nerve has three branches: ophthalmic, maxillary, and mandibular, and its maxillary branch innervates the ciliary mucosa. When your tears drain into the sinus cavity, they do not just passively sit there. The fluid contains sodium, potassium, chloride, and a mixture of proteins that create a specific osmolarity. The trigeminal nerve endings in the mucosa are sensitive to changes in osmolarity and temperature. Tears are slightly hypotonic relative to nasal mucosa. They have a lower salt concentration, and this osmotic difference triggers a depolarization in the trigeminal afferents. The signal travels to the spinal trigeminal nucleus in the brainstem, and from there it projects to the periaqueductal gray and the thalamus. What happens next is a phenomenon called diffuse noxious inhibitory control, or DNIC, a mechanism where one painful stimulus inhibits the perception of another pain elsewhere in the body. The trigeminal activation from the tears in your nose acts as a counterstimulus. It is not painful in the classic sense. It is a sensation of pressure or fullness, but it is strong enough to engage the descending pain modulation system. Dr. Didier Paré in Paris has shown that nasal stimulation with a cold saline in rinse similar in osmolarity to tears produces a measurable increase in pain tolerance across 3 minutes, measured by electric light within 3 minutes, measured by electrical stimulation of the sural nerve. The effect lasted up to 10 minutes after the rinse was removed. And this is where the third mechanism connects back to the first two. The trigeminal activation also potentiates the vagal response. The spinal trigeminal nucleus sends excitatory projections to the nucleus tractus solitari, the same brainstem region that receives vagal afferents. So when tears drain in your nose, the trigeminal signal amplifies the vagal signal that was already initiated by sobbing. The total effect on descending pain inhibition is greater than the sum of its parts. But there is a fourth mechanism now coming into view, and it is purely chemical clearance. Your emotional tears contain elevated levels of cortisol, the primary stress hormone. Cortisol is known to lower the pain threshold. Higher cortisol correlates with greater pain sensitivity. By excreting cortisol through your tears, you are physically removing a molecule that amplifies pain. Dr. Frey's original analysis found that emotional tears contain 2.1 micrograms per deciliter of cortisol, significantly higher than the 8 micrograms per deciliter found in reflex tears. Over the course of a crying episode, you can excrete enough cortisol to convix enough cortisol to measurably lower its concentration in your bloodstream. A study from the University of Tübingen in 2022 measured salivary cortisol levels in 6 participant session, and after a 10-minute crying session, they found a 44% reduction in cortisol within 20 minutes of the crying episode. And this reduction correlated with a significant increase in pain tolerance as measured by a pressure algometer applied to the index finger. So you have 4 mechanisms operating during a single crying episode. Opioid absorption from the nasal root, vagal activation from the sobbing pattern, trigeminal counterstimulation from the tears in your nasal cavity, and clearance of cortisol from the blood. Each one independently reduces pain. Together they create a window of analgesia that lasts somewhere between 15 and 30 minutes after the tears stop flowing. Slow. There is 5th mechanism now being mapped, and it involves prolactin. Emotional tears contain prolactin at roughly 1/8 times the concentration found in reflex tears, according to Dr. Frey's original 9981 data. But the role of prolactin in pain modulation was not understood until decades later. Prolactin receptors are densely expressed on the cell bodies of trigeminal nociceptors, the same nerve endings that detect the presence of tears in your nasal cavity. When prolactin binds to its receptor on these neurons, it triggers a signaling cascade that involves the enzyme protein kinase C and the inhibition of voltage-gated sodium channel. The result is a desensitization of the neuron. It becomes harder to activate, and when it does activate, the signal transmitted is weaker. A breakthrough study from Dr. Sarah M. Smith at the University of Texas Health Science Center in 2004 demonstrated this directly. They cultured trigeminal ganglion neurons from rats and applied prolactin at concentrations equivalent to those found in human emotional tears. The neurons showed a 35% reduction in the magnitude of voltage-gated sodium currents within 10 minutes of exposure. When the same researchers injected prolactin into the yeast rats, they observed a 40% increase in paw withdrawal latency on a hot plate at Hinate Tides Mainsin Semides Hidutik Nendiades Tri Rutisa. The prolactin effect was distinct from the opioid effect. When they co-administered naloxone to block opioid receptors, the prolactin analgesia remained intact. This is an entirely independent pathway. So, the prolactin from your tears enters your nasal cavity, crosses the mucosa, binds to receptors on the trigeminal nerve endings, and directly dampens the sensitivity of those neurons. This reduces the perception not only of the physical sensation of the tears thems, but also of any other pain signals, which we traveling traveling the trigeminal systems includes most of your pain, headache, pathways, and refer to pain from the sinuses. This may explain why people with chronic migraine often report a reduction in head intensity after a crying episode, though the clinical research on this connection is still in its earliest phase. The prolactin mechanisms also affects the hypothalamus. Prolactin crosses the blood-brain barrier through receptor-mediated transport at the choroid plexus, and once inside the brain, it binds to prolactin receptors in the paraventricular nucleus, the same region that releases oxytocin and vasopressin. Oxytocin is itself a powerful analgesic, a 27 Neuroscience by Dr. Neuroscience. Dr. Karen Lem MacDonald showed that intranasal prolactin increases pain tolerance during a cold press test by an average of 28% compared to placebo. So the prolactin you cry out is also triggering a secondary release of oxytocin inside your own brain, amplifying the pain relief cascade. Now consider the timing. Prolactin absorption through the nasal route takes up roughly 5 to 10 minutes to the cerebrosticon trains in the cerebrospinal fluid, based on work by Dr. William H. I.M. Born at the University of Lübeck, who measured the kinetics of intranasally administered peptides in human volunteers. That timing matches the typical delay between the peak of sobbing and the onset of noticeable relief. The prolactin and the leucine-enkephalin are working on different receptors, the opioid receptor mu subtype and the prolactin receptor, but they converge on the same behavioral output. Reduction in pain perception, a reduction in pain perception that is both subjective and measurable. And this is the layer of the research that I find particularly compelling. 5 separate mechanisms: opioid, absorption, vagal activation, trigeminal counterstimulation, cortisol clearance, and prolactin-mediated desensitization, all triggered by a single behavior that most people do not associate with physiology at all. The body has designed a redundant, multi-pathway system for pain relief that activates every time you cry. It is not a symbolic release. It is not a catharsis metaphor. It is a coordinated pharmaceutical manufacturing and delivery system. And you are the one producing every molecule, every nerve signal, every absorption event. Now I want you to consider the evolutionary context. Why would a mechanism, this complex 5-independent pathway converging on pain reduction, be wired into the act of crying? Pain is not the only signal your body needs to manage when you are in emotional distress. Social rejection activates the same brain regions as physical pain, the anterior cingulate cortex and the anterior insula, a finding first demonstrated by Dr. Naomi Eisenberger and Dr. Matthew Lieberman at UCLA in 2003 using fMRI scans. When a participant was excluded from a virtual ball-tossing game, their anterior cingulate cortex lit up with the same intensity as if they had been burned. The overlap is so precise that acetaminophen Tylenol has been shown to reduce the emotional pain of social rejection in a double-blind study published in 10 Psychological Science in 2010, led by Dr. C. Nathan DeWall. The same drug, the same dose, the same brain regions. So, if emotional and physical pain share neural circuitry, it makes biological sense that your body would deploy entire analgesic arsenal during emotional crises. A crying episode is not a breakdown in function. It is a biological response to a state of acute threat, the threat of social isolation, of overwhelming emotion, of a situation your nervous system cannot resolve through action. When you cannot fight or flee, your body activates the immobilization response. Crying is part of that response. And in that state, your body floods itself with molecules that reduce the immediate sensation of pain, the same molecules that would help you endure if you were physically wounded and alone. There is a study from the Weizmann Institute of Science in Israel, published in Science in 2021, that adds another layer. Dr. Noam Sobel and his team collected emotional tears from female volunteers watching a sad film, then placed a small amount of those tears under a small amount of volunteers through the nostrils of male volunteers who had never seen the film. The men were then asked to rate the sexual attractiveness of photographs of female faces. The men who had been exposed to the tears rated the faces as significantly less attractive, a 43% reduction in perceived sexual arousal compared to men exposed to saline. The effect was not due to smell the tears were odorless, but to a chemical signal that crossed the nasal mucosa and altered brain activity in the regions, including the hypothalamus and the amygdala. The tears were communicating information, not through emotion, but through emotions. This finding has profound implications. It suggests that emotional tears carry a molecular signal that other individuals can detect, absorb, and respond to. The same molecules that reduce your own pain may also be altering the physiology of anyone close enough to receive them. The signal is not a cry for help in the auditory sense. It is a chemical broadcast that lowers aggression, reduces sexual arousal, and promotes social bonding in those who are near. Your pain relief mechanism doubles as a social cohesion mechanism, and it operates below the level of conscious detection through the same nasal absorption pathway that delivers leucine-enkephalin and prolactin into your own bloodstream. Now the evident calibration. The opioid content of emotional tear has been replicated in at least 4 independent laboratories since Frey's original 1981 study. The vagal activation during sobbing has been confirmed by heart rate variability studies at multiple institutions, including the University of Sao Paulo and the University of Maryland. The trigeminal counterstimulation is well established in the pain literature for other nasal stimuli, though not yet tested specifically with tears. The prolactin mechanism in trigeminal neuron has been shown in animal models, but not yet in humans with emotional tears. The social chemical signaling from tears. The Sobel study has been replicated once by a group at the University of Duisburg-Essen in 2006, with similar results. So the evidence is strong as the opioid and vagal pathways, moderate for the trigeminal and prolactin pathways, preliminary but provocative for the social signaling. This is how science refines itself. Each study adds a brick, and the architecture of the explanation becomes more solid with every replication. Here is the part that I find poignant. Every person reading this, every person who has ever cried has produced, absorbed, and responded to a suite of molecules that are chemically identical to drugs currently being tested in clinical trials for pain management. You have access to a multimolecular analgesic system that costs nothing, requires no prescription, and activates spontaneously in response to emotional states. But the system only works if you allow it to complete its cycle. If you stop the tears, if you suppress them, if you tighten your throat and blink them back, you interrupt the drainage into the nasal cavity. The molecules are produced but never delivered. The vagal activation is initiated descend pain modulations to reach the descending pain modulation threshold. The cortisol remains in your bloodstream. The prolactin sits in the tear film on your cornea, never reaching the trigeminal receptors. You have manufactured the drug and then thrown it away. You can measure this effect. In 2006, a team at the University of São Paulo led by Dr. Marcel Beagle recruited 40 participants who reported chronic tension-type headaches and asked them to keep a daily diary of crying episodes for 8 weeks. They found that individuals who reported at least one crying episode per week on the visual analog scale and reduction of 10, the visual analog scale, compared to compared to weeks when they suppressed tears. The participants who suppressed tears for 4 consecutive weeks showed a measurable increase in pericranial tenderness, as measured by a pressure algometer applied to the temporalis and postli and trapezius muscles. The suppression did not just remove the analgesic benefit. It appeared to sensitize the pain system over time. What is happening inside the nervous system when you hold back tears is a sustained activation of the sympathetic fight or flight response without the sympathetic by the parasympathetic vagal activation. You are locking the body in a state of alert. The periaqueductal gray receives a persistent input from the amygdala signaling threat, but because the sobbing pattern never initiates, the descending pain inhibitory pathway is never triggered. The result is a net increase in pain sensitivity. This has been observed in a controlled laboratory setting by Dr. Rainer Kress at the University of Erlangen-Nuremberg, who asked 50 participants to watch sad film while immediately tears to watch a sad tears, or allowing them to cry freely. Those who suppressed showed a significant increase in galvanic response, a measure of sympathetic arousals, and a corresponding decrease in pain learned on a cold pressor test, administered 20 minutes after the film ended. The free crying group showed the opposite pattern, reduced sympathetic arousal and increased pain tolerance. This is where the cultural dimension inserts itself. In the United States, adult men cry an average of 1-4 times per month, according to a 2008 survey by the American Psychological Association, while adult women cry an average of 5-3 times per month. The same survey found that men report higher average pain sensitivity scores on the McGill Clinic what condition, a gap that had been attributed to hormone difference, but may also has been attributed to hormonal difference, and also a reflect mitris suppression of titling or suppression of the crying response. A study from the University of Painritelli published in Pain in 2020 and found page undergoing knee replacement surgery. Before the surgery and who reported rarely or never crying before surgery required snicklin dosi, hucheri significant higher dories 2-3%, more morphine equivalents on average compared to men who cried can or cried occasionally. The researchers controlled for age, body mass index, and baseline pain scores. The effect persisted, but there is a second, more subtle consequence of chronic suppression. The trigeminal nerve endings in the nasal mucosa can become sensitized to the absence of the chemical signals they expect. When prolactin and leucine-enkephalin are repeatedly produced but never delivered, the prolactin receptors on the trigeminal neurons may upregulate in an attempt to compensate, leading to a state of receptor hypersensitivity. When tears finally do drain, the response is more intense, but the base state between episodes is one of heightened vulnerability to pain. This phenomenon, called receptor supersensitivity, is well documented in other neurotransmitter systems, most classically with dopamine D, cascade with dopamine D2 receptors in patients treated with long-term antipsychotics. It has not yet been studied directly in the trigeminal system of chronic cry suppressors, but the pharmacology predicts it. Now here is the historical anchor. Dr. William Frey, the Minnesota biochemist, who first quantified the difference between emotional and reflex tears, was initially met with skepticism from his peers. The idea that tears could carry specific pain-modulating molecules seemed too elegant to be true. A romantic notion dressed in lab coat. Frey's 981 paper in the journal Ophthalmology was rejected twice before it found a home. Reviewers argued that the elevated protein levels could be explained by contamination from nasal fluid or by the longer film on the cornea during the emotional crying episodes. Frey responded by designing a collection protocol that minimized exposure time and by analyzing the tear sample for a blood protein that would pertain in data contamination. The protocol worked, the results held. Today, over 3 peer-reviewed papers have examined the molecular composition of emotional tears, and none have refuted Frey's fundamental finding that they contain unique pain-modulating molecules at elevated concentrations. The human story here is not about a single discovery, but about the architecture of the discovery itself. Frey was not looking for pain relief. He was trying to answer a question that had bothered him since medical school. Why do humans produce tears in response to emotion when no other mammal does? The leading hypothesis at the time was that emotional tears served as a social signal, a visible display of distress that elicited help from others. Frey did not reject that hypothesis. He simply added another layer. The tears all serve the individual who produced them. They are not just a signal, they are a medicine. The social signal and the self-medication operate on the same time scale, from the same source, by the same delivery route. The body does not have to choose. It does both at once, and this is the layer that the clinical pain community is only beginning to integrate. The concept of an endogenous analgesic system that activates automatically during motion stress, without tonically during emotional distress training, without equipment, without cognition, is not currently part of standard pain management curricula. But it could be. Dr. John D. Lozer, a pioneer in pain medicine at the University of Washington, wrote in a 2010 editorial that the failure to acknowledge the crying analgesia link represent a gap in our understanding of the body's own resources. He noted that if a pharmaceutical company had developed a molecule that simultaneously opioid receptors, enhanced actate, enhanced vagal tone, triggered diffuse noxious inhibitory control, cleared cortisol from the bloodstream, and desensitized trigeminal nociceptors all through a single, non-invasive, self-administered route, it would be considered a blockbuster analgesic candidate. The clinical trials was cost hundreds of millions of dollars. The patent would be worth billions, and yet the molecule already exists. It is produced by your lacrimal gland. The delivery route is your nasolacrimal duct. The dose is regulated by your emotional state, and the only thing preventing you from accessing it is the cultural instruction that tells you to stop. Now consider the practical implications of this for anyone living with chronic pain. The pain research community has known for decades that emotional modulate pain intensity and state modulate pain intensity. The phenomenon of stress-induced analgesia is well documented in both animal models and human studies. But stress-induced analgesia is generally understood as a fight or flight response mediated by endogenous opioids and endocannabinoids released during acute threat. Crying adds a distinct and additional layer. It is not stress-induced analgesia. It is cry-induced analgesia. The body enters a different physiological state, not the hyperarousal of fight or flight, but the parasympathetic dominance of the immobilization response. The heart rate slows, the blood pressure drops, the vagus nerve takes command, and in that state, The pain-modulating molecule flood the nasal cavity. This is not a recommendation. This is a description of a biological system that already exists. Whether or not you choose to use it is a personal decision shaped by your environment, your culture, and your history. But knowing that the system exists, knowing that your body has a self-contained analgesic delivery network that you can activate, simply not suppressing, ne suppresses and mul a natural changes the frame. The question is no longer why do we cry? It is why do we stop ourselves from doing something that our own physiology has designed to reduce pain. And there is a final layer that connects back to the very beginning of this discussion. Remember the fingertip at the inner corner of the eye. You touched the punctum. You felt the small ridge of tissue that marks the entrance to the nasolacrimal duct. Every time you cry, that duct fills with a fluid that contains molecules designed to cross your brain and in your brain within minutes. Every time you suppress those tears, you are actively blocking a delivery system that your body activated in response to a perceived threat. The tears are not the problem. They are the solution that your body pulled from its own pharmacy. The research that remains to be done is extensive. The role of specific tear components in different pain condition versus neuropathic pain versus visceral pain has not been mapped. The optimal duration of crying needed to achieve maximal analgesia has not been established. The interaction between chronic pain conditions and the cryings, whether individual with fibromyalgia or complex regional pain TREM or complex regional, cry differently or absorb tear components differently, remain unexplored. But the foundation architecture is clear. The mechanisms are identified. The pathways are named. The evidence, while incomplete, supports a conclusion that would have seemed improbable 40 years ago. Your tears are a drug delivery system, and you are the pharmacist. One last measurement. The average emotional crying episode in adults, according to data from Dr. Ad Vingerwitz at Tilburg University, lasts between 6 and 10 minutes. In that time, your lacrimal gland produces approximately 1.5 to 2 milliliters of tears. That volume contains approximately 30 nanograms of leucine-enkephalin of prolactin, roughly 6 picograms of prolactin, and enough cortisol to reduce your serum cortisol concentration by an average of 22% over the following 20 minutes. The total analgesic effect, measured using the cold pressor test in Dr. Balde 2008 study, corresponds to a 22% increase in pain tolerance and comparable to the effect of a single 5 milligram dose of acetaminophen. But without the hepatic metabolism, without the need for oral absorption, and without the delay of gastrointestinal transit, your body built this system over millions of years of evolution. It is older than language. It is older than culture. It runs on the same chemistry that connects a mother to her infant, that bonds a social group, that signals vulnerability without words. And it activates every time a tear forms in the inner corner of your eye. The next time you feel pressures building behind your eyes, you will know what is waiting on the other side that release. Not a symbol, not a catharsis metaphor, a coordinated pharmaceutical-grade analgesic cascade produced by your own body, delivered through your own ducts, bound for your own pain receptors. You are the pharmacy. The tears are the prescription, and all you have to do is let them drain.